Okay, we are on time. Welcome everyone. Let's get it started. This will be our last seminar for the semester, and then we'll resume on January. Today we have Dr. Benedict Albensi, Professor and Chair of Department of Pharmaceutical Sciences at Nova Southeastern University. Thank you, Dr. Ben, for accepting our invitations and agreeing to present here today. We're really excited what you do. To tell a little bit more about today's speaker, he obtained his PhD in neuroscience from University of Utah Medical School in 1995. As a postdoc, he developed MRI methods for investigating brain injury. Recently, the Albensi lab has shown early deficits and sex-based differences in mitochondrial function before the appearance of plaques and tangles. He has been ranked in the top 1% worldwide by the expertscape.com for his number of publications in past decades. He's a very accomplished researcher. I encourage all the attendees to read his bio and then try to collaborate with him. It's a very interesting research going on. Again, thank you again. Please take it away. The time is yours.
Okay, great. Yeah, thanks, Dr. Sapokota, to inviting me today. And it's really my pleasure to be introduced to the Florida Space Institute to talk more about Alzheimer's disease and hopefully how this could potentially help astronaut health. So hopefully you can hear and see me okay. These are my disclosures. And here's my agenda for the next 40 minutes or so. So what I want to do first is to look at some space stressors. I know all of you in this group have a strong command on this. But one big stressor that I'm interested in, of course, is aging, and especially accelerated aging. So there's many stressors on Earth and in space that are thought to accelerate the normal aging process. But for space travel, what we should imagine, of course, is spending months traveling to Mars, making great history, and then imagine coming back to Earth with a body that's aged faster than normal, and it's more prone to developing disease. And that's what accelerated aging is all about. And we're talking about accelerated aging due to space travel and not political service, of course. But accelerated aging seems to affect the heart, blood vessels, bones, the muscles start to degenerate really about 10 times faster in space than natural aging. And white matter in the brain also ages faster in space. So overall, there are many studies that have been showing that age seems to be accelerated, especially at the cellular and molecular level. So if this is true, then it makes sense that we think very carefully about how we age, especially in protecting our health in general, and in particular astronaut health. And so in my network, my community, we talk about hallmarks of normal aging. And that's what I show in this slide here. We can focus really on 9 areas, 9 hallmarks that are focused on cell and molecular changes in normal aging. And this includes deregulated nutrient sensing, epigenetic alterations, altered signaling, intracellular signaling, mitochondrial dysfunction, and the list goes on. And I circle a number of these in red here because these are areas that my lab works on. So accelerated aging really appears to be caused by a variety of space stressors and also challenges that appear to be mostly expressed at the molecular level. So in my seminar today, I really want to discuss how Alzheimer's disease research on Earth has the potential to help us understand and hopefully prevent some of these molecular stresses that occur during space travel and on the ISS. So here's some of the major health-related challenges that we know of for space travel. And I know all of you are familiar with these. These include not only the obvious risks like radiation and microgravity, but also the effects of being in a confined environment and sleep loss, and also being so far from home, especially with potential travel to Mars. So collectively, these conditions can affect multiple organ systems like the bone, the heart, muscle, the immune system, and of course the brain. And so just like the title of the NPR radio show called Hidden Brain, it might be a lot harder to actually see many of the changes in the brain as a result of space travel. So I want to look at some of the overlap now between space medicine research and my Alzheimer's disease research. So with regard to work in my lab, we study many of these space health related challenges that I just showed you in the last slide, and I list them again here. And many of these are conditions that affect the brain. And I highlight my focus really in the yellow highlighting. So in particular, we work on cognitive and behavioral changes, such as memory loss and brain connectivity changes. And we also study immune system changes and inflammatory processes, and then other factors that seem to affect aging. Now, most of these changes occur also in Alzheimer's disease. But what is Alzheimer's disease? Some of you might not be that familiar with this. So Alzheimer's disease is a type of dementia. It's the most common type of dementia. It's also listed as a neurocognitive disorder. And the typical symptoms in Alzheimer's involve impairments in memory. But there's also other anatomical, structural, and behavioral changes in cognition that are seen as the disease progresses. So in 1906, Dr. Alois Alzheimer's in Germany, of course, during an autopsy of a patient that he thought had mental illness, found these abnormal deposits and described these as amyloid beta plaques and tangles. And this was the very first time this was characterized in his patient, August Dieter. Now, there's been an intense focus over the last 30 years by neuroscientists and physicians and others that have looked at the amyloid hypothesis. And I'm not going to go into that great detail about that today, but we've had a lot of disappointment with developing new drugs that target amyloid, but that's really the topic of another seminar. So the factors I showed you, are they really what causes Alzheimer's disease? Well, we don't know for sure, but aging is actually the greatest risk factor for Alzheimer's. And it should be remembered, though, it's multifactorial. So what I mean is that it's really a combination of both environmental factors and genetic factors that we're trying to really understand for the cause of agents and how this disease progresses. So in addition to amyloid beta and tangles from chemically modified tau, there's other mechanisms and processes that we think play key roles in Alzheimer's, especially inflammation and mitochondrial dysfunction. And these are two things that we study in my lab. So now this is how your brain might look after 20 years or so as a result of Alzheimer's pathology. And you can see several really noticeable changes. You see the enlarged ventricles, you see overall shrunken cortical volume, you see the cell loss, so many different structural changes. And as a result of cell loss, of course, we get altered neural networks. And when these networks are altered, then we have changes in brain connectivity, and that's going to ultimately affect behavior and cognition. So let's talk a little bit now about immunity and inflammation and how this is affected both in Alzheimer's and space travel. So one central molecule I've been studying for, I think, 25, 30 years now is nuclear factor kappa B, or NF-kappa B. Now, this molecule regulates a number of aspects of innate and adaptive immune responses. It's also a mediator of inflammation. So specifically, NF-kappa B is a transcription factor, which means it's a specialized protein. And once it's activated, it's used to turn genes in the nucleus on and off. Okay, so you can see it's composed of three different parts or subunits: the I-kappa B, the P50, and P65 subunits. And this is just one combination. There are other variations as well, which helps in its selectivity and also helps to generate a variety of different functional roles. But turning the right genes on or off is critical to both healthy aging and optimal health. So David Baltimore, of course, the Nobel laureate at Caltech, first described NF-kappa B, where he focused on this molecule in the transcription of immune cell genes. So in humans, NF-kappa B has also been implicated in Alzheimer's disease. And you can see in the confocal images on the right, the colored red and green slides there, we see the co-localization of P65 subunit of NF-kappa B with actual amyloid beta plaques. So this has been found in human brain in the Alzheimer's disease condition. So there's also a molecule I want to point out that's amyloid precursor protein, or APP. And this molecule is important because when the molecule is cleaved a particular way with certain enzymes, Then it generates this fragment, this amyloid beta fragment that many people talk about, this sticky toxic protein that many people believe is a causative agent. So APP also, this molecule also appears to be a gene target of NF-kappa B. So NF-kappa B is really the center of the universe, at least from a biochemical signaling point of view. And this slide shows potential activators of NF-kappa B. So some of these include oxidative stress, cytokines, infectious agents, and so on. It's even involved in memory. But factors that stimulate the immune system also activate NF-kappa B. So there's a variety of different responses that occur once activated, and this includes even more cytokines. So there are a number of feedback loops that occur, inflammatory responses, and the turning on and off of hundreds of specific genes and various networks. So speaking of the networks, of all the pathways studied in the space environment, the NF-kappa B network appears to be commonly affected across many cell types. So in this slide is showing a couple different databases that were looked at that shows the regulatory relationships of NF-kappa B. One was obtained from the TranFact database, and the other one's the Metacore analytical suite. So these are both knowledge bases that are connected to and looking at inflammatory networks. So based on the data in these two databases, the regulatory network of NF-kappa B consists of 54 total target genes and 77 transcriptional regulatory interactions that are all implicated in inflammatory responses. So the crucial genes of the NF-kappa B regulatory network were identified in this database in this study to really reflect the consequences of inflammation. So alterations in this network, the NF-kappa B network, are associated with a host of age-related diseases. So this really could be a primary concern for astronauts in long-term spaceflight. So also, in addition to the inflammatory changes in the brain, we also know that it can affect osteoblasts, and these are the cells that form new bones. Also, cardiac muscle changes related to muscle atrophy occur from this sort of network activation. So these are many of the things that occur to astronauts on the ISS and during spaceflight. So I think that these processes are important to look at more carefully in astronauts. Now, studies have shown astronauts experience immune system dysfunction. There's a number of papers that have been published over the years, and this includes the reactivation of latent viruses. And so a latent virus, of course, is a virus that's present in the body, but exists in a resting state without producing more virus until there's some trigger. And 95% of all of us have latent viruses, and they don't become activated again until there's a stressor. And certainly spending time on ISS has shown that these latent viruses are again triggered, which can have long-term consequences, especially on longer-term space flights. So some of the triggers that we found so far include radiation and microgravity, but there might be others too that are related to immune system changes that we haven't quite identified yet. Now here's a table from a review that I looked at. It shows a list of papers where NF-kappa B was studied in models of microgravity, and some of these models were on Earth, but then some of the studies were also on the ISS. So these models included a combination of either cell lines or, in some cases, human and also mouse models. So many of these studies here in this table found that there were increases in NF-kappa B and also changes in the expression of NF-kappa B gene targets, those genes that NF-kappa B can turn on or off. So in the same paper, there's another list, another table that showed associations between radiation and NF-kappa B activation. So overall, NF-kappa B activation seems to increase as a result of radiation and in a dose-dependent manner, which is important. So now I want to just talk a little bit about some of my work and other work with sex differences, especially how that connects to mitochondrial function. And the mitochondria, of course, are the powerhouse of the cell, the organelle that produces ATP, the cellular energy currency. So what I should tell you, though, just to give you some background, is that there are sex-based differences in the occurrence of Alzheimer's disease, and we can't yet fully explain this just by longevity alone. So in other words, we know that women on average live longer than men, but that by itself doesn't explain why we see these sex differences. And about 2/3 of women in the US and Canada, or I should say 2/3 of those with Alzheimer's in the US and Canada actually are women in terms of who is subjected to Alzheimer's disease. Now, it's interesting because women are at lower risk for Alzheimer's as compared to men before menopause, but their risk triples for after menopause. So some scientists believe this is due to this decrease in sex hormones as we age. And of course, males and females age differently. So in males, there's only a gradual decline in testosterone during aging, but in women, there's a very rapid decline in estrogen just after menopause, and that estrogen molecule continues to decline with aging. So there's a profound difference here in what way we age, especially in our middle ages and as we get older. And interestingly, A-beta deposits are also quite different in their deposition in females and males, and we don't know quite why that is yet. So to this end, we've looked at sex differences in my lab in our studies of Alzheimer's, and you can see I pointed out here too the instruments that we use to assess mitochondrial function in some of these tests. And in the upper right corner, right here, is the Seahorse XF24. This is a plate reader system. You can put a plate in of 24 different wells, and you can either use cell cultures or homogenates and test mitochondrial health. And then the other system that we use is this Oxyograph Ouroboros 2K. This is a nice system. This is a more, this isn't as user-friendly, it's more laborious, but this can be used in clinical trials, and one can actually test human white blood cells and look at mitochondrial health using this instrument. So using both of these instruments, we published a number of papers over the last few years on these sex differences, and overall, we found many sex-based differences in mitochondria when comparing our male versus female Alzheimer's disease transgenic mice. Sex differences also exist in space, and most of you probably know this already. So, for example, in mice, male mice exposed to energetic helium ions show higher rates of neuroinflammation, and female mice have shown neurocognitive effects also related to radiation. With regard to female astronauts, women have greater loss of plasma volume, and women astronauts also experience differences in heart rate and vascular resistance as compared to male astronauts. And the list goes on. There's quite a few differences, and there's still quite a bit to learn as well. So one important thing, and what I'm leading to, of course, is space travel can also disrupt mitochondrial dysfunction or mitochondrial function, which we see in Alzheimer's disease, and we see it in many age-related conditions. So given this, a variety of different mitochondrial markers are studied by myself and others to ascertain mitochondrial health in various contexts and conditions. So one of these that's actually been used on the ISS, one marker includes so-called cell-free circulating mitochondrial DNA, or CCF mitochondrial DNA. So this is a nice marker because it's used, what they are, they're short sections of mitochondrial DNA. And it's released from cells that are undergoing stress or some other damage or pathological event. So it's a nice marker to assess mitochondrial health. What you may or may not know is that mitochondrial DNA is actually all inherited from our mothers. So the entire mitochondrial DNA molecule is through a maternal lineage, which is very interesting. So there's a lot of markers to look at, as you can see in this table. And in my lab, we measure many of these markers just as a standard procedure. So for example, when we look at so-called mitochondrial biodynamic changes, we look at one marker called DRP1, or dynamin-related protein 1, and this is a mitochondrial marker for mitochondrial fission. So to our surprise, when we started doing some of this work using Western blotting techniques, and that's what we're looking at here, to measure protein expression, we started finding these sex differences at very early ages. So you can see that we tested 3 different age groups. So we started finding sex differences at 2 months and 6 months of age. So overall in this study, and we've published this data if you want to look it up or learn more, but we showed both age-dependent and sex-dependent differences and changes in DRP1. So overall, we found more fission, okay? So fusion and fission processes occur in mitochondria oftentimes when they experience some sort of metabolic stress or environmental stress. So also, we looked at other markers. So we looked at the mitofusion proteins. These are regulators of fusion in the mitochondria. And again, we found age-dependent and sex-dependent changes in the expression of these proteins. So why am I telling you all this? Well, fission is needed to create new mitochondria, and it contributes to so-called quality control. And it allows us to remove the damaged mitochondria, and it can facilitate programmed cell death or apoptosis during high levels of cellular stress. Whereas the fusion process actually helps to mitigate stress by mixing the contents of partially damaged mitochondria as a form of complementation. So disruptions in these processes not only affect development, but they've been implicated in neurodegenerative diseases like Alzheimer's disease. So overall in our mice, when we look at these markers, we see changes in DRP1 and MFN1 and 2, and we find a shift towards more fission. So changes in markers of mitochondrial function, really, one of the reasons I'm telling you this is because they can have important consequences, not only for mitochondrial health, but also for memory. And so I'm hoping that you see this video here. It's a very short video clip that I'm showing. And in this video, mitochondria have been fluorescently labeled that can be seen moving to sites of high energy demand. In other words, to sites of synaptic plasticity. Can you see that?
Yes, we can see the video.
Okay, thank you. So this movement only works, though, if all the cellular machinery is in place. So that is the mitochondrial proteins, some of the ones I already mentioned, but there are others too. And so these proteins are all involved in biogenesis and biodynamics in the normal cell and in the normal neurons. So memory takes energy, and mitochondria move to these sites of high energy demand. So we also look at another marker, uncoupling protein, UCP4. This is actually involved in heat regulation. And likewise, we found its expression levels change in an age-dependent and sex-dependent manner. So disruptions in these markers, and these are used as a mitochondrial solute carrier, can affect the membrane potential, the mitochondrial membrane potential. And it also leads to reactive oxygen species or ROS levels that can change, which will induce oxidative stress. So overall, similar studies have been attempted in space. And for example, omics databases have been collected and published. And this is a terrific study led by Afshin Beheshti, who heads up one of the NASA Gene Lab team. I've been interacting and learning more about the Gene Lab for over a year now, and they're really producing a lot of really cool data. And this was actually published about 3 years ago. But overall, in this study that was published in Cell, they found 3 important things. They found that spaceflight overall affected isolated cells more than whole organs. They also saw that the liver went through a lot of types of different gene expression changes. And with regard specifically to mitochondria, they found that the mitochondrial stress response pathway was induced. And this was very exciting for someone like myself that studies mitochondrial biology. So evidence of this altered mitochondrial function and mitochondrial DNA damage was found in samples taken from urine and blood in astronauts in this study. So it's very similar to what we see in Alzheimer's disease with regard to the mitochondrial stress response, if not identical. And one molecule that they talk about in this paper that really caught my attention is a molecule called PGC1 alpha, which I'll talk a bit more about in just a few minutes. But this is a very important molecule for mitochondrial regulation, for biogenesis, for mitophagy, a whole host of mitochondrial functions. And they found alterations in this study published by Beheshti. So PGC1 alpha is really a master regulator of the mitochondrial life cycle. So overall, this data was taken from a variety of astronauts, and it also incorporated data from the NASA twin study and looking at mitochondrial stress. So let's talk a little bit more about memory and some of those studies now. So memory impairments, of course, are a hallmark of Alzheimer's disease, type of dementia. We don't see memory impairments in all types of dementia, but we do in Alzheimer's. And so in my lab, we use a variety of different methods for testing memory in humans, in our clinical trials, and also in our mouse or rat animal models. So the top 2 panels on the high left are cognitive instruments we use in our clinical trial in Canada for testing memory in humans, right here and here. This one here is called the MOCA, or the Montreal Cognitive Assessment Test. And this has been used because it's been found to be highly sensitive to MCI. And MCI stands for mild cognitive impairment. And this is the stage right before mild Alzheimer's disease. So we use this in our clinical trials as a screening tool. And so it has several different domains that are measured, such as visual, spatial, attention, verbal, and math abilities. The other tests that we use, there's several that we use. I'm just going to describe two. This is another test that we use here. It's called CANTAB. This was developed at Cambridge University in England. And so this is another neuropsychological test, a cognitive test, where you can see a variety of different left or colored squares and circles. So with CANTAB, it's a very short test, and we can measure things like attention or psychomotor coordination or motor speed or reaction times and spatial working memory. And these are all things that astronauts, of course, have to be good at. They have to be able to have that attention span, and they need to have strong spatial working memory because they have so many tasks to carry out on the ISS and just during space flight in general. So some other tests that we do in our animal models, here on the top right, the Morse water maze and the Barnes platform. These are both mazes that are used for rodents, either mice or rats. And we use these to measure potential changes in spatial memory. And in fact, it's hippocampal dependent spatial memory. So we also do something called LTP, electrophysiology, long-term potentiation. I show that in these two middle slides. So really all it is obtaining brain slices, keeping them alive. It's an experimental paradigm for memory encoding. So we can measure voltage changes in these hippocampal brain slices as another assessment of plasticity and memory. And then finally, we use PET scanning and MRI in both our human trials and in our animal models, either to look at functional changes like that are produced by PET scanning, or to look at structural changes that can be evaluated with magnetic resonance imaging or MRI. So we can measure things like glucose uptake and brain metabolism, and also changes in brain area region or I should say area or volumes, which will change structurally as the brain starts to get damaged.
Okay, hi Ben, we have a questions before we go to the next slide.
Yeah, go ahead. Yep.
So at the beginning of this, of the previous slide you were just on, you mentioned the diseases having memory impairment, but you made a distinction between Alzheimer's and dementia as far as memory impairment goes. So does memory impairment in this case, is that referring to memory recollection or memory storage or creation, or both?
So
and the two diseases?
Yeah, it takes a while to explain because there are various forms of memory. We have episodic memory, remembering events. We also have other intrinsic or implicit memories and explicit memories. There's emotional memory. There's short-term memory and long-term memories. There's even intermediate forms of memory. Many biologists are very sloppy about actually using those terms correctly. The psychologists seem to be a bit more precise about it. But with Alzheimer's disease, we do see this impairment in the conversion of short-term memories to long-term memory. So it takes days to weeks to months to fully consolidate a memory. And to consolidate a memory, there has to be rehearsal. And there also has to be a period of sleep, because most reconsolidation occurs during sleep. So you got a lot of moving parts. You got a lot going on here. So I'm not sure if I'm addressing your question, but there are, so that with Alzheimer's disease, it is this impairment in short-term memories because they don't get converted to long-term memories. And long-term memories are primarily stored in many neocortical sites. Okay, where the hippocampus is a smaller structure, and that's where many memories start. And it takes days to weeks for those memory traces to fully consolidate and then transition to other cortical storage sites. Is that what you were wondering?
That's actually a little higher level than but yeah, okay. I did follow most of that. Yeah, thank you. And that is what
Okay.
I was.
But I guess the point in dementia is that, Alzheimer's disease, it involves memory, and that's quite clear. But you have other types of dementia, like Lewy body dementia and frontal temporal lobe dementia. You don't always get memory impairment. You get some impairments with executive function. And in fact, one of my friends from college developed frontal temporal lobe dementia. She wasn't having memory problems, but she was having problems adding and subtracting. So it's very interesting. And sometimes hallucinations and delusions are other features of other types of dementia, but Alzheimer's is the most common type. Probably about 60 to 70% of all types of dementia is what the Alzheimer's type is.
Thank you.
Sure. So I want to talk a little bit about something else in neuroscience that we refer to as the so-called default mode network. This is really fascinating to me. So the DMN helps us understand not only memory, but brain connections. And so the integrity of this default mode network appears to be crucial for memory and mental health in general. So what happens in Alzheimer's disease is that we have disruptions of this connectivity, like I implied earlier, because you lose cells, you lose neural networks, and eventually this connectivity fails. And we see this within brain regions that are associated with the default mode network. And there are certain circuits in brain regions that form this network that I'm not going to go into today. But cognitive decline in Alzheimer's disease is reflected by this decreased signal complexity in these DMN nodes. And that's what they did in this study. So in this study, they compared these nodes and they compared the normal patients on the left, and they compared how these nodes and the complexity of connections changed in the Alzheimer's patients on the right. So it's another tool that neuropsychologists can use and neuroscientists to look at how the brain's changing from pathology. So this is also, there is the potential to use this in spaceflight and with astronauts too. And the reason would be because a variety of studies have indicated that space travel can affect memory and brain connectivity. So why is that though? What's causing it? So one reason is the levels of carbon dioxide, and some estimates that carbon dioxide on the ISS are 10 times higher than what we experience here on Earth. Radiation also, and I think most of you probably know more about this than I do, but 1 week on the ISS is like a year's exposure on Earth, or 30 times more. And microgravity, of course. Microgravity can induce fluid shifts in the brain. So this is important, and this is really noticeable when the astronauts come back. And all of this together can affect memory and cognition. And of course, sleep deprivation. So I don't know if a lot of astronauts complain about this, but on average, astronauts only sleep 6 hours. And what we just talked about a few minutes ago is the fact that memories are reconsolidated during sleep. So if you don't get enough sleep, you're not going to really reconsolidate and form those permanent memories. That's number one. Number two is that amyloid beta protein, that toxic sticky protein that I told you about, that's generated from one of the types of cleavages of that APP molecule, that's actually cleared during sleep. So if you don't get enough sleep, you're not clearing the A beta. So it's a healthy mechanism. And so sleep deprivation is a double whammy for both accelerating amyloid deposition and also interfering with memory reconsolidation. And then finally, just the effects of long-term confinement and isolation is a concern. And in fact, some studies have shown that it might reduce neurogenesis. And neurogenesis, when I was in college 100 years ago, we didn't know that neurogenesis exists. So now we do. And there's two primary sites, the hippocampus and the olfactory bulb. So in other words, nerve cells can divide. And so on spaceflight and on ISS, it seems that confinement is actually somehow connected to reducing neurogenesis. And I think that this is probably a factor of many, many things, but this one particular study was pointing that out. Now, here's one study that was done in space over 6 months, but before they went to on the flight, functional MRI or fMRI was obtained, and then they did it again after the flight. So they did a variety of experiments here looking at spatial working memory, like we talked a few minutes ago, and how this affected brain connectivity. And so they found overall that the working memory stayed pretty much the same, but they were able to show that there was a change, the modification in the connections. So the brain circuit connections were disrupted in some instances just from 6 months of time up on ISS. All right, so I want to move over to countermeasures now. I think I'm running out of time, but try to get this done in 40 minutes. So I'm part of a team that's working on ways to grow and transfuse mitochondria. So we're doing a lot of work with our mitochondria and Alzheimer's, but we started doing something different a couple of years ago. So we're looking at applications. It's a team I belong to, and I'm looking at the Alzheimer's stuff, but we've got a couple investigators at Stanford that are looking at the transfusion of mitochondria in the retina. We've got a team up at Laval University in Quebec that are looking at activated mitochondria from platelets. We also have some people in Kentucky looking at sepsis and so on. So all of us together are collecting this data on mitochondrial transfusion. And so that's one thing I'm going to talk about in just a minute. So another Alzheimer's Association team that I'm on, we look at diet and brain metabolism and its role in improving memory and cognition. I'm not a nutrition expert, but I am interested in the changes, the positive changes that can be achieved in the mitochondria from a proper diet. In some cases, we're looking at new drugs or repurposed drugs, and I'll show you a couple examples of that in order to prevent pathology. And then finally, we spent a lot of our time looking at sex-specific therapeutics. In other words, more personalized medicine, especially for mitochondrial dysfunction and memory. So here's one of our team members presenting our work over the summer, a couple months ago, with mitochondrial transfusion. This is a partnership with this company called Matrix. And so this is the overall idea, is that we can extract stem cells and then isolate healthy mitochondria from humans or animals. The mitochondria are then grown in a bioreactor, and this can create larger quantities. The healthy mitochondria are transfused into the animal or the patient, which would increase the number of mitochondria. So the idea is that by increasing the number of healthy mitochondria, we should be able to weaken the effects of the damaged mitochondria. So we started doing some experiments along these lines. And so my lab published this paper last year in mice. And basically, and that's the study right here. So I guess I'll show that data in a minute. And then our team also published this other paper, and this just came out a couple months ago in both animals and in humans, looking at mitochondrial transfusion. And as we speak, we're looking at the possibility of testing these ideas in larger animals. And in fact, we're going to start doing some of this work in Toronto in a facility that houses dogs. So I mention all this because it stands to reason that if we can show benefit from mitochondrial transfusion, that this would have great impact on astronauts on long duration spaceflight that could benefit from a transfusion to help reduce the effects of immune dysfunction, or even on human colonies on the moon. So this could be an effective countermeasure. In fact, here's some data from this slide from my study that we just published. And what we're doing here is looking at Western blots. And basically, we're comparing the expression of protein subunits at 1 month of age compared to mice that are 12 months old, okay? So we looked at two different regions. We looked at the cortex and the hippocampus. And then what we found is that you can see all the black bars here are expression levels of mice. These are proteins in the electron transport chain from complexes 1 to 5. And we could find that over 12 months of aging, that in every complex, in every case, the expression of those key subunit proteins diminished, okay? It's quite clear. So why is this important? Well, complexes like complex 4, it's the site of oxygen consumption, and complexes like complex 5 is the site of ATP generation, okay? So here's the next figure in the paper. So then what we did is that we took mitochondria from liver, from 1 month old animals, and we injected these healthy mitochondria using IV administration but into 12-month-old animals. And we were able to enhance one of the mitochondrial complexes, and that's complex 2. So you can see this is what it was like without any transfusion, and we tried a couple of different doses, and so we were able to improve the protein expression at complex 2 using this method. So we were pretty excited about that. Now, there's a lot more work to be done. This is just in a mouse model of aging, and of course, the next step is to do this in Alzheimer's mice and to use a variety of doses. So there's a lot of things yet to do. And of course, to try these techniques in larger animals. So in the next set of experiments, I'm just going to show you the SOP that we use that to modulate mitochondrial function. We use this with the XF24, that one instrument I showed you a few minutes ago. And it's called the mitochondrial stress test. And we add a variety of reagents in an ordered fashion to stress out the mitochondria. So that's the method that we use. And so this data took, I don't know, a year or two to collect. And you can see that what we're looking at is that we're using a different mouse model. These are 3XTG Alzheimer's disease transgenic mice. We're comparing males to females, okay, different males to females. And then we're sampling the hippocampus versus the cortex. And then we're looking at 3 different age groups. So 2 months, 6 months, and I think 14 months of age. So the main measurement is OCR, or oxygen consumption rate. And that's what happens at complex 4 that we just talked about a few seconds ago. So what we found very quickly is that we found everywhere there's a red arrow shows a set of experiments, not just one experiment, but it's really kind of an average. So we found that in females, the OCR deficit was much earlier than males. And we found a different pattern when you overall compare males to females as the mice aged, okay? So in addition to the sex-based differences, we found that there's age dependence and that the cortex seemed more susceptible to mitochondrial changes than the hippocampus. This corresponds to what we see in the human when the entorhinal cortex is actually affected first in Alzheimer's, and then the hippocampus is affected later in time. We published this in the paper Cells. So this is some work done by my PhD student in Canada. In this set of studies, we've been testing sex hormones. And we're doing this to follow up on the observations I just showed you in the last slide. So we're testing both male and female sex hormones to see if they're neuroprotective, number one, and then also to look at a variety of different biochemical pathways that are involved in glucose metabolism, brain metabolism, and also memory encoding. So we found so far that the female sex hormone estrogen is actually effective in improving several key signaling pathways, including this PGC1 alpha pathway that Ashvin showed in that Cell paper with the Omics Gene Lab study a few minutes ago. So there's a variety of common metabolic pathways we've been identifying that are implicated in Alzheimer's disease in this particular study, and also in space. Now, here's another study where we actually worked with creatine. Now, creatine, these mice were fed a creatine diet for 8 weeks. Creatine is an over-the-counter product you can buy at the grocery store, or you can get it from the food you eat, like meat and fish. And it's also used by bodybuilders because it can rapidly build up muscle, okay? Something that astronauts will lose after being on the ISS for a while. So the idea here is to see if there are protective, especially neuroprotective effects of creatine, so to prevent neurodegeneration, and also to see how it affects memory and brain metabolism. So overall, to make a long story short, we found creatine was protective, and we also found that it actually inhibited the NF-kappa B pathway that I talked about at length a few minutes ago. So we published a couple papers that are listed here at the bottom. So I think diets rich in creatine could actually be used for astronauts during spaceflight as another potential countermeasure, because they have a positive effect against this NF-kappa B induced inflammation. And there's also benefits such as improving muscle mass. And there's also data showing that it can improve memory, okay? So if you want to learn more about NF-kappa B's role in the mitochondria, I refer you to this review paper that I published. Now, here's a TV interview that I did last summer that was announcing my clinical trial, and this was with flaxseed. And only recently did we conclude the study, so I can't tell you all the results. But the rationale for doing this is because we wanted to see if flax can improve memory. And that's because flaxseed contains omega-3 fatty acids, okay? So there's been a lot of research at my old research center in Canada that showed flax is beneficial for heart health, but no one had looked at it for brain health. So the primary outcome was to look at memory, but we're also looking at blood samples to see if we can reduce inflammatory markers. And we're also using PET scanning to see if there's any changes in brain metabolism or any structural changes using MRI. Now, flax contains omega-3 fatty acids similar to fish, and fish containing omega-3 have been found to have many, many beneficial effects for improving physiological function, right? Well, there aren't any fish on the moon, but perhaps we can grow flax in space. So some people have done some work with flax related to the space program. And so people have looked at flax for space travel, as you can see by some of the studies that I point out here. Flax can actually inhibit NF-kappa B activation, and it seems to have effects on immune and inflammatory responses in general. Also, some have tried to grow flax in microgravity. So it is possible that growing and consuming flax on the ISS or even on moon colonies during space travel could be another countermeasure for boosting astronaut omega-3 fatty acid levels. Now, one last drug I want to talk about is this anti-cancer drug leukemia, or nilotinib. This was done by one of my postdocs, Ida Adlimigadam, and we published this in the journal Aging and Disease, okay? So nilotinib is a tyrosine kinase inhibitor. It's used to treat leukemia. And of course, I'm sure you know that radiation exposure has been shown to cause leukemia. So this is very interesting because nilotinib has been found to have benefit also in Lewy body dementia and Parkinson's dementia. And my colleague at Georgetown University in Washington showed this in clinical trials. So Scott and I have been working on this to see how this works. And so we've tested this drug in our mitochondria. So this is very interesting because a repurposed drug, it's been around for 13 years, has a very good safety record. So there's a lot of data and evidence out there already on nilotinib, so it's well understood. So that's another rationale for using this drug. So Scott's still testing this in Alzheimer's patients in the memory clinic, and we've been testing it in our mouse models, and we've shown that it has an effect on improving mitochondrial function, it improves the OCR, and it also improved complex 4 expression that I told you about, that important complex that's the site of oxygen consumption. We also found that it actually reduced amyloid and tau in our Alzheimer's transgenic mouse model. And then finally, we have evidence to show that it has a positive effect on PGC1 alpha signaling, that one molecule I keep harping about. In fact, in some of our Western blot experiments that I show here, we show increases in a number of molecules, including PGC1 alpha and NRF2, which is another good guy that we found in our 3X Alzheimer's disease mice, okay? So now, given that nilotinib has this positive effect on leukemia and also on Alzheimer's disease pathology and also enhances mitochondrial function, and it regulates important signaling pathways, it seems like this drug would be perfect to put in a pharmacy toolkit for the ISS or any long-term space flight, okay? So the other thing I just want to talk about, and then I'll wrap this up. So with regard to looking at A beta and tau generation in space, the results have been mixed, which is surprising. So in one study with 3X mice, the same mice that I use, they found no change in amyloid or tau markers. But in a study by five Russian cosmonauts who spent 169 days on the ISS, they actually showed several different changes. They found changes in amyloid beta, they found changes in tau, and they found changes in some other markers that are indicative of Alzheimer's disease pathology. So this report was in JAMA Neurology. So that was very interesting. So now here's a timeline showing several changes related to Alzheimer's disease pathology. And the current thinking is that if amyloid can be cleared earlier on, then perhaps we can prevent Alzheimer's. But not all scientists agree with this view. But this is the current view, and this is the approach that many of the drug companies are taking, okay? So right now there's 3 drugs that are used to enhance cholinesterase or to enhance cholinergic function that I show here. There's another drug called lamantine, and these are 4 drugs that have been used for over 10 years to try to combat Alzheimer's disease. Well, none of these drugs work very well. And really they work by different mechanisms. This drug works to block the NMDA receptor. And then there's 2 new drugs. These were just released. One was just released in January 2023, and another drug was released in 2021. So they work by different mechanisms, but how they work is different than the older drugs. They actually clear amyloid, or they block amyloid. So this might be interesting. I don't want to get into all the details, but there are different forms of amyloid, and these drugs will clear different forms of amyloid differentially, okay? So why am I telling you all this? Well, in case one wants to perhaps test this for long-term space travel, we might want to ask, does it make sense to use these two drugs as a preventative measure? Well, I don't think so, because there's some serious side effects with these drugs with regard to brain swelling and brain bleeding. And so I don't think they're the best choice for testing on astronauts. The other thing is that these new drugs clear amyloid, but they have little or no effect on memory and cell loss, which is pretty important. All right, so let me just wrap it up. We really have a lot to learn, and I think we have a lot to share for both the Alzheimer's disease community and the space community. The ISS and space travel, I think, provides a unique environment, as all of you know, not only for astronauts, but to advance what I'm interested in with regards to research related to Alzheimer's that has profound effects for those on Earth. And so specific changes in sex differences, brain connectivity, changes in memory, mitochondrial dysfunction, we share all these mechanisms and processes that I think both communities want to study. And I also believe that nutritional interventions like creatine and certain drugs like nilotinib that focus on enhancing mitochondrial function really should be considered as potential countermeasures for a long duration spaceflight. And then finally, enhancing immunity, I think, is an important countermeasure as well for protecting brain function. And to this end, this NF-kappa B signaling pathway that I talked about and the PGC1 alpha pathway are both emerging as pathways that we should be studying more to target and to reduce the effects of accelerated aging and space travel. And this is just showing you how some of these molecules connect together. If you want to read more about that, here's the reference here. So taken together, I think these findings really support a role for nilotinib that could be exploited for space travel. So I want to thank all my staff in Canada. I apologize for going over time, especially three of my postdocs, my postdoc fellows, Dr. Ida Lumigadon, Dr. Snow, and Dr. Dvorževic, that produced a lot of the data I showed you. Also my new staff, my new team I'm forming here in Florida. Dr. Mohammed Sabir produced the video that I showed you. And then these are my collaborators in the US and Canada that help to collaborate on these projects as well. So I think I'll just end there. This is some of my funding over the years and my new funding from the Alzheimer's Association and the NIH. So thank you for listening. I'm happy to answer questions in the few minutes that we have remaining.
All right, thank you, Ben. We're still in time, so no issue with that. I think we're open for questions. Julie, go ahead.
Hi, Ben. This was very interesting. I was wondering if, if when the astronauts come back, there is a sort of like, what's the state of affairs? Is the damage done, or can the body recuperate, basically?
That's a good question. And I think the answer is that it depends. It depends on the system we're talking about. And there seems to be a huge difference between 6 months in space and 1 year in space. It seems like there's a lot of recovery possible if we only spend 6 months in space. But for some of the, I think in the NASA twin study, I think that one astronaut was there for at least 12 months. And so some of the effects might not be as reversible. So this is a concern. But again, it depends on the organ system that we're talking about. So many of these effects are on the cell and molecular level. But we do start to see some of the structural changes, especially in the brain, especially as we get closer to that 12-month point.
Thank you.
Okay, any other questions for our speaker? For online participants, if you have questions, either raise your hands or unmute yourself. Okay, I think I have one question. So for a healthy body, how much of a risk of the body carrying like latent viruses that are potentially bad? Like,
Can you repeat?
really carry? those
the first part?
viruses for generations?
Yeah, I mean, like I said, if I heard the question correctly, you're asking how much of a risk are latent viruses?
For our body,
It-
yeah.
It's a huge problem. So, I mean, we learned a lot recently from COVID. And, you know, some of these effects can be long lasting and the triggers are very interesting. So, as I said, I think a few minutes ago, 95% of us have latent viruses. And once the immune system is weakened or triggered by some pathogen, that's when they express themselves. So, you know, the way viruses work, of course, is that they insert their DNA into our DNA. So they're pretty much with us the rest of our lives. So the older we get, the more influence viruses can have on us because they've had time to insert their molecular constituents into our DNA. So it's a real concern. And I think, you know, I think that's another consideration, of course, are vaccines before any astronauts are going to be going to the ISS and or on long-term space flight missions or colonies on the moon. Varicella virus is, I think, the most studied one so far for space travel.
Okay, thank you. Any more questions? Yeah, it looks like we do not have any other questions, and let's thank our speaker again. Yeah, thank you, man. It was a great talk.
Okay, well, thank you for inviting me again, and nice to meet folks at the Florida Space Institute.
All right, thank you,
Thank you,
everyone.
Ben.
We'll resume next semester.
Okay, bye-bye.
Bye.